Hello, in this video we're going to be going over how to read and put together Punnett squares. This video was edited and put together with iMovie for my W200 production badge. So our question, what is a Punnett square? The Punnett square is a diagram that is used to predict an outcome of a particular cross or breeding experiment. And an example of a Punnett square is right here. And we'll go over how to read these, how to put them together, and all of that. But this is what they look like. This is a Punnett square. So we're going to hit a couple major points when talking about Punnett squares. First, I want to go over phenotype and genotype. So a phenotype is what you can physically see in a So for example, eye color, skin color, being tall, being short, things like that. Those are phenotypes. A genotype is what you can't see. So it's kind of hard to explain, but a genotype is, so let's say you could have got a tall gene from your mom and a short gene from your dad, and you only see the tall gene. But you have deep down inside a short gene that you got from your mom. It's just not expressed. It's in your genes and you can't see it. You can't physically see it. So therefore it's a genotype. I've highlighted physical see and phenotype because that's how you can define phenotype. If you can physically see it, it's phenotype. If you cannot physically see it, it's genotype. That makes it pretty black and white and easy for you to differentiate. Now we're going to go over to dominant and recessive genes. A dominant gene is expressed by a capital letter in the Punnett square. As we've seen before, there's capital letters, there's lowercase letters. For a dominant gene, if the gene is present, then it will be expressed in the phenotype. So if a capital letter is expressed in the genotype, you will see the capital letter in the phenotype. So for example, if we have a capital T and a lowercase t, you put them together, you have capital T, lowercase t, and you get a tall person, because no matter what, it only takes one uppercase letter for it to show in the offspring. Recessive genes, if this gene is present, it will take two for it to be shown inside the phenotype. So it takes two recessive genes to be shown inside the phenotype. For example, if you have the two T's, it'll take a little t and another little t for the offspring to be short. Now we're going to talk about how to fill out a Punnett square and how to read one. So if you look over here, we are mixing a capital T and a little t with a capital T and a little t. Let's just say the top one is the mom, and this one over here is the dad. Okay? So the mom is capital T little t, the dad is capital T little t, so they're both the same. But when they mix, they create a variety of phenotypes and genotypes. So the mom's tall and the dad's tall. In order to fill this out, you take the T and you drag it down. So you have capital T, you put a capital T here, and you put a capital T here. And you go over, there's a little t. You put a little t here, and a little t here. And you do the same when you're taking it across. So instead of going down, you're just going to go across. So you have a capital T and a capital T. Here you have a little t and a little t. And if you think about what we said previously with phenotypes and genotypes, for a phenotype to be shown, it only takes one capital letter. So let's say this is tall and short. This child's going to be tall, and this child's going to be tall, this child's going to be tall, and this child is going to be short. And I know that's kind of weird because both parents are tall, and you're like, how do they create a short offspring? That's because they both have a recessive allele. They have little t in their genotype. It's not expressed in their phenotype, but it is in their genotype. And if it's in your genotype, it's possible for it to be passed down to your children. And this does not guarantee anything. So all their kids can be short, all their kids can be tall. It's not like the first kid's going to be tall, second kid's going to be tall, third kid's going to be tall, fourth kid's going to be short. It doesn't go like that. It's the chance of it. So here there's a 75% chance that the kid's going to be tall and a 25% chance that the child is going to be short. And that's because there's 3 with dominance. So 3 over 4, that translates to 75%. And there's 1 out of 4, 1 over 4 translates to 24%. Here we have an example with flowers. So in this example, we're going to consider that capital B, the dominant gene, is purple. And the recessive gene, 2 little b's, will result in a white flower. So if we look here, we'll wait for it to restart so that I can walk you through it. They drag both them down, they drag them across. There goes the B, there goes the B, there goes little b, there goes little b. Big B goes across, big B goes across, little b, purple, 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 white. And the reason it's white is because once they drag the both little b's down, it had 2 little b's, so therefore that's white. All these were purple because they had a big B, so the dominant shows in those. And that is Punnett squares.